My name is Julie Theriot. I'm a professor of biology at the University of Washington in Seattle. My group studies primarily cellular biophysics, and we're mostly interested in understanding how human cells are able to move and to respond to external signals. The study of the organization of cells is interesting and important in part just because cells are the most complex objects that build themselves that we know of. Looking around this incredible building that we're in right now, you can see the hand of the architect who thought what it should look like and designed the experience of walking into this amazing courtyard, looking upwards and seeing all these beautiful structures. Looking inside of a cell using an electron microscope or a light microscope, you can see an organization that is at least as complex, or in reality much more complex than what we see in this building. And yet there was no architect who decided what it was supposed to look like. Instead, all of the individual components of the cells come together on their own, interact with one another in order to build these incredibly elaborate structures that are also dynamic. So it's as if this building were able to take itself apart into just separate little bricks and then rebuild itself every few minutes for decades. This sort of complexity in self-organization is something that we don't really have analogs to understand at the macroscopic scale. It's something we can really only understand by studying cells themselves. And everything that happens in living organisms depends on cells being properly functional. As you go about just your normal life as a human, you probably don't spend a lot of time thinking about the processes that all the individual cells are doing in your body. But you wouldn't last for any amount of time if they weren't all doing their jobs exactly right. And this includes all the different cells of the body, including cells of the immune system that are able to fight infections or repair damage that happens immediately so that you can just continue onwards. It includes the cells of your gastrointestinal system that are able to digest food and then absorb nutrients from your food. Cells of the blood that are able to carry oxygen around and also carry those immune cells everywhere in the body so that they can fight infections wherever they appear. And every single one of these cells is as complicated not only as a building but as a whole city, but incredibly rapidly dynamic, changing all the time, and again somehow able to figure out how to do all of these incredibly complicated things that they have to do without there being any directions for them to follow. They just have to figure it out for themselves. So understanding the principles of self-organization at the cellular level is, I think, one of the most profound challenges in modern biology. We now have techniques that we can identify all of the individual proteins, all of the individual macromolecules inside of a cell, but we're just beginning to understand how they're actually able to come together at these larger scales in order to achieve the functions that cells are able to achieve. One of the really amazing things about pathogenic bacteria is that they're excellent cell biologists. That is, they understand the inner workings of human cells much, much better than humans actually understand our own cells. And this is particularly evident in bacteria like Listeria monocytogenes that are able to invade human cells and then cause the components of the human cells to rearrange in ways that are beneficial for the bacteria. So Listeria, for example, triggers assembly of the host cell cytoskeletal protein actin in order to generate these beautiful comet tails that actually enable the bacteria to move around inside of the cells. And if they weren't able to persuade the human proteins to rearrange in this way, the bacteria wouldn't be able to get anywhere. They would infect a cell and then just remain there until the immune system killed them off. But by hijacking the cytoskeleton of the human cells, the Listeria are able to spread around inside that cell and then also spread from one cell to another, and even spread, for example, into the brain of an infected human patient that they wouldn't be able to do if they couldn't use the human proteins to do that. So one of the things that's really useful about studying how the bacteria do this is it helps us understand the principles of organization of the human components, because the bacteria are able to trigger them at will. And it's possible to think about these bacterial diseases really as being not something that the bacteria are doing on their own, but rather a dialogue between the bacteria and the human cells that they infect. And if we can just cut off the communication, if we can just somehow prevent the bacteria from telling the human cells what to do, or stop the human cells from paying attention to what the bacteria want them to do, then an infection could be stopped just dead in its tracks. One particularly interesting example of self-organization, and particularly the dynamics of self-organization, is in the context of cell motility. So cells, many cell types are able to crawl around on their own. And this can either be part of normal biology, like in the context of the immune system. If you get a little cut in your skin, then white blood cells actually come out of your bloodstream and crawl towards the site of the cut in order to clean up any bacteria that might have gotten in through that breach. However, also it's possible for cell motility to happen in the context of pathology, and one of the most important examples of that is in cancer. So the most common cancers are carcinomas, that is epithelial cells that become cancerous. And if they just over-proliferate but stay in place, that can be treated surgically. Cancers, most cancers only become life-threatening after metastasis, which is when the tumor cells start leaving the site of origin and actually crawling through human tissues, crawling into the lymphatic system or crawling into the bloodstream, and then traveling around the rest of the body in order to disseminate lots of small tumors everywhere. And once the cancers reach that stage, it's very hard to treat. So part of the impetus for wanting to understand how cells move and how cells crawl and how cells choose the directions that they go in is both to be able to prevent processes like metastasis, where cells are able to crawl places that we don't want them to, but also to be able to enhance processes like immune reactions, or even in the context of both, many modern cancer treatments depend on what are called CAR T cells, where cells of the immune system have been engineered to attack cancers inside the human body. And understanding cell migration then is important both for understanding how to prevent the cancer from spreading, but also for understanding how to maximize the ability of those engineered T cells to find the cancer and destroy it. So right now we're really still focused on understanding the mechanisms of cell motility at this very mechanistic level, what the proteins do that actually enable cells to move, how force is generated that gets them from one place to another. But the eventual hope, the ambition, is to be able to understand those processes well enough that we can control them, that we can either increase them in the case of immune cell reaction or decrease them as in the case of cancer metastasis.